Spiders are one of the most successful predatory arthropods alive today. These 8-legged hunters are the 7th most diverse animal order on Earth, beaten only by beetles, flies, wasps, moths, true bugs, and snails. More than 51,000 species are recognized, meaning they make up over 15% of all described terrestrial predators. Spiders are also one of only 3 lineages of arthropods to have evolved the capability of producing silk for prey capture, alongside certain mites and some larval insects. Spiders, known scientifically as Araneae, from the Greek for spinner, have some amazingly diverse hunting strategies, with web builders, ambush predators, and active hunters. They're found on every continent except for Antarctica, occupying habitats from desert caves to tropical rainforests. Unlike centipedes and scorpions, spiders subdue prey through venom injection, with hollow fangs connected to specialized glands, whereas their arachnid cousins rely primarily on mechanical crushing or stinging. Despite their astonishing modern diversity, the origin of neurotoxic venom in spiders is still poorly understood. The early evolution of this spectacular chemical arsenal is one of the most intriguing mysteries in arachnology. Some species have developed venoms so sophisticated they can selectively target specific nerve channels, essentially turning an animal's own nervous system into an instrument of its capture. For starters, what exactly makes spider venom so remarkably effective at subduing prey? Well, that brings us to the first fascinating aspect of spider neurotoxicity. The most striking feature of venomous spiders is their ability to inject complex cocktails of neurotoxins that interfere with nerve signal transmission. These creatures are equipped with chelicerae, paired appendages bearing hollow fangs, that allow them to deliver venom directly into prey tissues with surgical precision. Large hunting spiders like the Brazilian wandering spider, Phoneutria nigriventer, possess fangs measuring up to 7 millimeters in length. These are just stunning adaptations. It's clear that this animal is perfectly designed for venom delivery, with those needle-sharp chelicerae for penetration, just as we see in successful predators. The venom glands themselves can measure up to 10 millimeters long in large specimens, storing substantial quantities of neurotoxic compounds. Phoneutria species are definitely capable of causing severe neuromuscular disruption, with potent venom and all the right biochemical components to pull this off. However, the key difference is the specific mechanism by which these toxins operate at the molecular level. Neurotoxic venom is a biological superpower. By binding to voltage-gated sodium channels in nerve cell membranes, these toxins can then prevent normal electrical signal propagation and cause uncontrolled nerve firing to induce paralysis or spastic muscle contractions. Spiders that use neurotoxic hunting strategies possess relatively large venom glands, part of the prosoma, that are involved in the detection of prey resistance and the calibration of venom dose. Research published in 2016 by the Instituto Butantan in São Paulo showed that Phoneutria venom contains over 150 distinct peptide compounds. This discovery helped explain how wandering spiders evolved such remarkable prey-subduing capability across diverse prey types, from insects to small vertebrates. The venom works by blocking specific ion channels, causing the prey's muscles to contract involuntarily while simultaneously preventing coordinated movement, essentially hijacking the nervous system's command structure. Looking at prey response patterns, this shows us how neuromuscular hijacking relates to successful prey capture, helping to solve questions that spider venom researchers had been debating for decades. High-speed videography studies also offer insight into how exactly paralysis progression works in these animals. Some modern wandering spider species retain relatively simple venoms, but Phoneutria species possess highly derived cocktails with multiple synergistic components. It therefore seems that Phoneutria uses its venom to simultaneously attack multiple neural pathways for maximum efficiency. So it's clear that venomous spiders have developed remarkable diversity in neurotoxin composition and delivery methods. These predators must have undergone significant biochemical adaptation, which makes sense as this would have coincided with the diversification of potential prey species during the Cretaceous period. But what about related species? The Sydney funnel-web spider shows us interesting parallels. While Phoneutria uses sodium channel modulators primarily, Atrax robustus has evolved calcium channel blockers, demonstrating how evolution can solve similar predatory problems through different biochemical pathways. These spiders are found across tropical and subtropical regions, from Central American rainforests to the Atlantic forests of Brazil. This widespread distribution shows how successfully neurotoxic hunting strategies have adapted to different prey communities. A 2019 study examining 47 different Phoneutria venom samples found that toxin composition varies geographically, with coastal populations showing higher potency against crustacean prey. This research, conducted by the Universidade Federal de Minas Gerais, revealed that venom evolution tracks local prey availability remarkably closely. As a quick aside, prey animals exhibit distinct behavioral phases during envenomation. Initial hyperactivity lasting 15-30 seconds, followed by progressive loss of coordination, then complete paralysis within 3 to 5 minutes depending on prey size and injection site. It's at this point that we must look at how modern neurotoxic spiders are classified to make better sense of these animals. The relationship of Phoneutria to other Catenidae has long been debated. They were previously positioned as relatives of wolf spiders, but recent research has found better genetic support for their position within the wandering spider family, sharing ancestry with less venomous hunting spiders. The classification of neurotoxic spiders splits them into major groups: the Mygalomorphae with primarily mechanical venom effects, and the Araneomorphae with more sophisticated neurotoxins. The Mygalomorphae include tarantulas and funnel webs, while Araneomorphae include wandering spiders, widows, and recluses. You'll remember earlier the distinction between different types of ion channel targeting. The recent discovery of amber-preserved spider fangs from Myanmar, dating to 99 million years ago, has been key to understanding venom evolution. These specimens show clear evidence of venom delivery channels and were found to be closely related to ancestral hunting spider lineages. A 2018 study looking at 200 different spider species investigated how venom complexity relates to modern lineages. According to this study, neurotoxic sophistication increased dramatically during the Paleogene period, coinciding with the radiation of mammalian and avian prey. So, to summarize current knowledge, neurotoxic spiders show remarkable biochemical diversity, have evolved multiple independent solutions to prey subduation, and are classified into several major groups based on venom mechanism and phylogenetic relationships. Now, I need to quickly add here that not all large spiders possess medically significant venom. Many tarantulas, despite impressive size, rely more on mechanical damage than neurotoxicity. So, neurotoxic spiders are remarkable predators that have evolved some of nature's most sophisticated biochemical weapons. From their precision venom delivery systems to their molecularly targeted neurotoxins, these animals represent one of nature's most successful predatory adaptations. The ability to essentially commandeer a prey animal's nervous system, forcing muscles to work against the animal's survival, is absolutely incredible when you consider the evolutionary refinement required. Today, these spiders can be found in diverse habitats worldwide, where they continue to hunt using venoms refined over millions of years. Understanding these creatures helps us appreciate not only predator-prey dynamics, but also offers potential medical applications, as several spider venom compounds are being investigated for pain management and neurological treatments. Recent research has revealed previously unknown venom components with highly specific ion channel targets, though questions remain about the full diversity of neurotoxic mechanisms across spider families. Future studies will likely uncover even more fascinating details about how these animals evolved such precise molecular tools for prey capture. There's clearly still much to be learned about neurotoxic spiders, but there's no doubt that this fascinating group of arachnids represents an astonishing example of biochemical evolution and predatory innovation. Pretty amazing when you think about it. Nature has engineered molecular keys that can unlock and manipulate the most protected systems in animal biology. I hope this has served as a useful overview of how spiders hijack prey nervous systems. Let me know in the comments what other topics you'd like to see covered. Please like this video and subscribe to the channel if you enjoyed it, and be sure to press that bell icon so you can learn more about our world and the wonderful life that surrounds us all.